Hello everybody, my name is Sabina Putri and I'm from Group C, going to explain about the interaction of plant cells and their environment. Firstly, what is plant cells? Plant cells are the basic unit of life organisms of the kingdom Plantae. They are eukaryotic cells, as we can see in the pictures in here, which have a true nucleus along with specialized structures called organelles that carry out different functions. And here we got some example of organelles like mitochondria, cell wall, vacuole, and chloroplast. The definition of environment is the surroundings or condition in which person, animal, or plant live or operates. It can be sunny, windy, cloudy, rainy, or snowy, or other environment that can be in around plant. So what are the interactions between the plant cells and the environment? What can the plant cells do in order to survive in different environments? The first interaction that we can get in here is photosynthesis. So the photosynthesis is the process by which green plants and certain other organisms transform light energy into chemical energy. Or photosynthesis, is the simple definition of photosynthesis is a series of plants that making food by themselves. And here we get two steps, that is light dependent reaction and light independent reaction. The first light-dependent reaction is to convert light energy into chemical energy such as ATP and NADP. And in here, we got some system to do. There is for system 1 and for system 2. But the first one that you can go through is for system 2, and then we get to electron transport chain, and then we can get to the for system 1, and then we get to the export transport chain. In the photosystem 2, is absorbed the best at 680 nanometers. Otherwise, in photosystem 1, it absorbs the best in 700 nanometers. And the process itself, it takes 4 process. There is electron excitation, is where when the electrons moving from one place to another place, and then this is the photolysis. The photolysis is when the water molecules break down into hydrogen and oxygen and electrons. After that is photophosphorylation. There's 2 kinds of photophosphorylation. The first one is a non-cyclic one and the cyclic one. The non-cyclic one is when the electrons are removed from water and pass through from the system, photosystem 2 and photosystem 1 before ending up in NADPH. But in the cyclic one is when the electron break this pattern and instead loop back to the first part of the electrons transport chain, repeatedly cycling through photosystem 1 instead of ending up in NADPH. The second step is the light independent reaction, or we used to say it a dark reaction. The process is took 3 processes, that is fixation carbon dioxide, reduction, and regeneration of RUBP. In the fixation stage, 1 molecule RUBP reacts to the carbon dioxide. As a result, 2 molecules of 3-PGA are formed. There's 1 phosphate and 3 carbons in H2-PGA. During the reaction, the number of carbon atoms remains the same despite the formation of new bonds. So this 3-PGA gains electrons, and that is why it is a reduction reaction. And after that, in regeneration of RUBP regeneration refers to the cyclical process where the photosynthetic enzyme Rubisco fixes carbon dioxide into sugars that fuel plant growth and productivity. And in here, we got the yielded product, that is 3GP, ADP, and ADP+. But in order to get the glucose is the further formation of 1 extra 3GP. 3 carbon atoms are needed, and consequently 3 turns of the Calvin cycle. Therefore, 6 rounds of the Calvin cycle needed for the formation of 1 molecule of glucose. The next interaction, there is respiration. Respiration is the process of the absorbing oxygen contained in the free air to produce air, carbon dioxide, and energy that plants need to grow and develop. There is 4 steps in this respiration, that is glycolysis, the carboxylation oxidative, the Krebs cycle, and electron transport chain. The first step is glycolysis. The glycolysis is the metabolic pathway that converts glucose into pyruvic acid. Then the yielded product in here is 2 ATP, 2 NADH, and 2 pyruvate molecules. And in here, we can see the picture of the step from glucose into the yielded product. There is 2 ATP, 2 NADH, and 2 pyruvate molecules. The second step is the carboxylation oxidative. Oxidation reacts in which a carboxylate group is removed, forming carbon dioxide. The product of this step is 2-acetyl-CoA, 2 NADH, and 2 carbon dioxide. So in the carboxylation oxidative, we have the 2-acetyl-CoA, and in here we get to use the acetyl-CoA. So the Krebs cycle is a series of chemical reactions to release the stored energy through the oxidation of acetyl-CoA. So therefore, we get the product of CNADH, 2 ATP, and 2 FADH, and 2 carbon dioxide. The last step in this respiration is the electron transport chain. Electron transport chain is an arrangement of protein complexes that exchange electrons from electron benefactors to electron acceptors by means of product redox responses, and couples this electron exchange with the exchange of photons over a layer. And the product yielded in here is ATP and H2O. The process in here is complex 1. Complex 1 is 2 electrons are carried to the first complex aboard NADH, and the complex 2 is directly received FHDH2, which doesn't pass through complex 1. And then the third complex is composed by cytochrome B under FES protein and its center and cytochrome C proteins. And in this complex 3, it carries electrons, not oxygen. And the fourth complex is composed of cytochrome proteins C, A, and A3. So there is 2 groups. There is 2 heme groups. There's 2 heme groups and 3 copper ions. And the last process is from chemoosmosis. Chemoosmosis is the free energy from the series of redox reaction just described is used to pump hydrogen ions across the membrane. The last interaction that we have in here is osmoregulation. Osmoregulation is the process of maintenance of salt and water balance or osmotic balance across the membranes within the body fluids, which are composed of water plus electrolytes and non-electrolytes. And in here, the stomata are important in regulating water loss through evapotranspiration. And on the cellular level, the vacuole is crucial in regulating the concentration of solutes in the cytoplasm. The next is we have the 3 conditions of osmoregulation. The first one is hypertonic. The hypertonic is when the plant cell loses water and hence trigger pressure by plasmolysis. Pressure decreases to the point where the protoplasm of the cell pulls away from the cell wall, leaving gaps between the cell wall and the membrane and making the plant cell shrink and crumple. And the second one is hypotonic. Will absorb water by endosmosis so that the increased volume of water in the cell will increase pressure, making the protoplasm push against the cell wall, a condition known as turgor. And acetonic is the cell is not plasmolyzed and it's not turgid either. The green parts of the plant will drop and are unable to hold the leaves up into the sunlight. And this is what it becomes in picture. In acetonic, it became flaccid. In hypotonic, it becomes turgid or normal, and in hypertonic, it's plasmolyzed. So example of the process of this osmosis is when the osmosis is how plants are able to absorb from water from soil. Roots of the plants have a higher solute concentration and the surrounding soil. So water flows into the roots. In plants, gourd cells are also affected by osmosis. There are cells on the other side of leaves that open and close to allow gas exchange. When the plant cells are full of water, the gourd cell swell and open stomata. Small holes that allow the plant to take in carbon dioxide are released and to take in carbon dioxide and release oxygen. Thank you. That is for the interactions of plant cells and environment. The next presentation is going to be Zidan.
Thank you, Shabina. My name is Zidan Rafli Einstein. I'm the second presenter of Group C, and today I'll be talking to you about the roles of plant cells in the production of chemicals/drugs. Plants has been used as a remedy to treat certain illnesses throughout human history. from the manuscript of the Unai to the Egyptian papyrus to ancient Chinese writings, has all mentioned the uses of plants as a medicine to treat certain diseases. Even in today's society, plants has a major role in certain people's healthcare, especially in third world countries where there's a lack of healthcare infrastructure. I'd like to start this presentation by defining the word of pharmacognosy. Pharmacognosy comes from the Greek word pharma means drug and cognosy means knowledge, the knowledge of drugs. In this plants, which it is defined as study of plants and other natural substances as possible sources of drugs, meaning it is the study of the active components inside the plant cells which can then be used. Why can plants be used as medication? What makes plants so special is because they contain phytochemicals. All plants produce chemical compounds which gives them an evolutionary advantage. These traits come from generations of evolution due to environmental changes. And because of this, it makes plants useful for human medicine. Phytochemicals are the chemical compounds produced by plants that gives the ability against the resistance of fungi, bacteria, and viruses, and they live within the normal metabolic activities of the plant. And so, phytochemicals can be found in the two types of metabolites. The primary metabolites, which are mainly fat and sugar, and the secondary metabolites, which are compounds that are found in a smaller range of plants serving a more specific function. For instance, toxins in plants which are used to deter predators, or pheromones which are used to attract insects for pollination. And it are these second metabolites which gives a therapeutic effect in humans, allowing these plants to be refined as drugs. So now I would like to talk about the different varieties of photochemicals, or which are the active ingredients of plant-based drugs, and gives the characteristics of treatment. Let's start with alkaloids. Alkaloids are composed of a wide variety of plants that contains nitrogen-bearing molecules that makes them very active. They are a bitter-tasting chemical, and there are several classes. There are several classes of different modes of action as drugs, both as for recreational and pharmaceutical. A great example is opioids, or in this case, in the bottom left, is morphine, which is used as a pain to treat pain. And yeah, second active ingredient is glycosides. You can split the glycosides into two categories, which is cardiac glycosides and cyanogenic glycosides. Cardiac glycosides are compounds found in various medicinal plants. Examples are foxglove and lily of the valley, and they have a strong action in the heart. An example is beside the morphine, it is a foxglove plant, which the compound dig, dig, digioxin can be synthesized from, and it is used for heart treatment. And cyanogenic glycosides are glycosides based upon cyanide, but in small doses, and they serve as a muscle relaxant. Uh, phytophenols has a diverse role in defending plant diseases and predators. Uh, they're mainly used as a hormone mimicking phytoestrogen and estrogen tannins, which has been administrated to be used for uh, gynecological disorders such as fertility, menstrual, and menopause. So they're mainly focused in female care. And terpenes are strong, aromatic, and serve to repel herbivores. Uh, their scent makes them useful for essential oils, perfumes, and aromatherapy. An example is thymol, which is seen on the right, due to its antiseptic properties, it can be used as a mouthwash. Plant drug discovery. First off, what is what is drug discovery? Drug discovery is the process in which potential new medicines are identified. It is a long, strenuous, research-based program where you basically try to find new drugs to sell. And so, right now, I've been talking to you for the past few slides. I've been talking to you about how plants can be used as medicines. But now I'll be talking about how we can identify these active compounds and those can be refined into drugs, and so we can reap the benefits. First off, what are the plant's roles in drug discovery? What can the plants be used in drug discovery? First off, the plants can be the main subject, as in the active compound from the plant be directly used as a drug. It can be the main subject of discovery, and so yeah. Second, active compositions may act as a lead compound. So lead compounds are compounds that has the ability to cause an effect, the ability to become a drug, but right now at its stage, it has to be modified for it to work. Basically means that it has the potential to become a drug, but at its stage right now it can't. And so active compounds from plants can be like the foundation of future drugs. It can be the composition, but again, it has to be modified first. Third, phytochemicals can be used as marker compounds for standardization of crude plant materials or extract. Marker compounds are chemical constituents uh, which can be used to verify its potency and identity. Basically, phytochemicals can be used to identify and identifies the crude plant materials, which is very useful. And last, phytochemicals can be used as pharmacological tools. So the ethnopharmacological approach is where scientists study the plant compounds from ethnic groups or traditional medicine. Basically, researchers would go to different ethnic groups, communities, sites where drugs has been documented from the prehistoric times, and they would learn the knowledge what these uh, what they have to offer. Uh, why scientists would ever do this is because they have a notion, assumption where active compounds isolated from plants are likely to be safer than those with no history of human use. Basically, plants that has been used for generations in history from ethnic groups are probably safer than those that from a plant that has no background, no knowledge whatsoever. And so it's mainly for safety, and also it's a step was it the scientists can build on the knowledge uh, from these ethnic groups, and so it will be much easier to start. And so the study involves the identification and the endotaxonomy of uh, of the natural materials from which these plants come from. The scientists will do a complete rundown where the composition of these plants and learn more about the plants. They'll also watch and understand and learn the traditional preparation of the remedy, because usually herbal medicines are done has to be prepared. Maybe there's a certain recipe you need to know, and so researchers will study that. And so moving on to my next point is the bio-evaluation of the possible active ingredient. Based on the information they've learned, they will make a judgment if whatever they are doing actually has an effect, or is this just a placebo. So these are how the study approaches, and a great example of where this approach has been successful is the drug development of the Rauwolfia serpentina, which now today is a safe, effective treatment for hypertension. HTS, or high throughput screening, is another method for to identify if a plant can be used as a potential drug discovery candidate. HTS is a drug discovery process where it allows automated testing of a large number of chemicals for a specific biological target. Due to the immense chemical diversity of phytochemicals, there is no researchers that has the manpower, money, or time to be able to test each phytochemicals from each plant. It it wastes time, and it takes too long. Through HTS, it allows researchers to conduct a testing on phytochemicals simultaneously in a much faster rate, saving them time and manpower. And so through this method, scientists can quickly identify which active ingredient works on a specific biological test. That's it for all. Thank you for listening to my presentation. I hope you enjoyed it. Thank you.